All right, so step one here, transcription. We're going to take the DNA that is inside of your nucleus and we are going to get a transcript, a copy of it, and that will be mRNA, messenger RNA, that will leave the nucleus and find a ribosome. Remember, the purpose of transcription is to carry that code of instructions out of the nucleus. DNA does not leave the nucleus, so proteins are made in the cytoplasm, whether they're made with the ribosomes that are connected to the rough ER, or if they're made with free-floating ribosomes inside of the cytoplasm. Transcription is going to end with the formation of this mRNA. So the process of transcription starts with RNA polymerase, which is a multi-complex protein that is going to bind to the DNA and it's going to unzip just the gene that needs to be copied. This process is efficient and very specific. There's a thymine, adenine signal that tells RNA polymerase where it's going to bind to the DNA and start copying. Then RNA polymerase is going to use complementary base pairing rules that are going to match the RNA nucleotides with the exposed part of the DNA nucleotides that are there. So once it kind of slips in right here and starts to unzip the DNA, that exposed DNA gene is going to be matched with RNA nucleotides. Remember that RNA is different from DNA in that it has uracil. So adenine is going to bind with uracil to create this RNA. And cytosine is going to bind with guanine still. And so once this process has finished, the DNA is going to zip itself back up and it's going to release this completed mRNA molecule. This mRNA molecule is now single-stranded and it contains a transcript of the original DNA gene. It also no longer is going to have adenine in it. So it'll have cytosine, guanine, thymine, and now uracil when it leaves the nucleus. So in this process, your DNA right here is unzipped and copied, transcribed into mRNA. It's printed out all funny. This mRNA, this single strand, is what's going to leave the nucleus. And then the DNA is going to zip back up, and this mRNA, a separate molecule, is a transcript of a gene that was there. I should also point out that DNA is the template, right? And RNA is the copy, which will be read as the directions. And RNA is made in this 5' to 3' direction. And the DNA template is read from this 3' to 5' direction. When you see these 5s and 3s, they're talking about the sugar, the way the sugar is directed. If it's a 5-point sugar, 3-point sugar direction, it's describing the actual just layout of the molecule or the direction it's read, because it is read specifically like in a sentence type of sequence. So just RNA is made in the direction of 5 to 3, and the DNA template is read in the opposite direction from 3 to 5. So if this is my DNA part right here that is unzipped, it's being read in this 3 to 5 direction, but the RNA is being made 5 to 3. So technically, this mRNA strand that's going to leave the nucleus is a transcript copy of the DNA that's in there, and the sentence structure of how it's read is in reverse. It's backwards. It's like a backwards copy. All right, I promise this will make better sense here in a minute. So for an example of how transcription would look, my DNA sequence beginning with 3' and ending with 5' is a gene. It's a gene sequence that is inside of the DNA molecule in your nucleus. And then polymerase is going to come along and transcribe the DNA molecule into a mRNA sequence. For every thymine, it's going to pair it with adenine, and for every adenine, it's going to substitute uracil. Cytosine still pairs with guanine. Guanine is going to pair with cytosine. Cytosine, guanine, thymine to adenine, but every A is going to turn into a uracil. And this molecule is in the reverse direction of the original template. And this 5' and 3' situation has to do with the sugar that's pointing out in that direction of the last nucleotide here. This process is so specific. There are modifications that can be used to double check, make sure it is accurate. RNA splicing is when mRNA has these introns. They're this non-coding region, and exons, which are the actual coding region. And this process removes introns and splices exons together. Basically creates a little different combination of exons, and thus you make multiple polypeptides from just the one gene. So it's almost like storing multiple gene codings in a small piece. That 5' cap, which is what we also call the G cap, it's a modified guanine type of molecule, and it's added to that 5' end, and that facilitates in binding the ribosome. So after transcription has taken place and the mRNA is floating around, out in the nucleus and the cytoplasm, these modifications can take place. There's also what we call a poly A tail, and that is added to this 3' end, and it helps that mRNA leave the nucleus. So I point out these modifications so that you know that just the transcribe portion, it's not the be-all, end-all. There are the things that modifications that can take place inside of the nucleus and then outside of the nucleus as it is traveling around the cell.